So starting with the carbohydrates, this is the information that is required in the study guide. So I've broken it up. Instead of having one big chart, I just have one line of the chart for each of the macromolecules. So the elements that we see in carbohydrates, they generally just have carbon, hydrogen, and oxygen. If it's a pure carbohydrate, simple carbohydrate, it's probably just carbon, hydrogen, and oxygen. And literally, this is carbon and hydrate. So if you look at this, it is carbon, and hydrate means water. So for every carbon, there is an H2O, and the hydrogen to oxygen is always in a 2-to-1 ratio. So the monosaccharides consist of glucose, galactose, and fructose. Those are monosaccharides. That'd be a monomer. Disaccharide is the dimer. Saccharose refers to sugar. And so that's what generally these are the sugars. And so you glucose, galactose, and fructose. If you add glucose to glucose, you get maltose. Glucose to galactose, you get lactose. Glucose to fructose, you get sucrose. So sucrose is that sugar that you put on your cereal. It's like regular table sugar, or just, regular sugar. And fructose is combined with glucose to make that. Polysaccharides are the polymers, and they include things like cellulose and starch and glycogen and peptidoglycan. So cellulose and peptidoglycan are cell wall structures, and in plants, starch will store the monosaccharides, will store glucose, and glycogen will store this in animals. And so those are some of the functions that they have. But generally it's sugar, it's about energy. Here's the other slides that have to do with carbohydrates. But basically when you see saccharose, that means sugar. Monosaccharide is a simple sugar that has like 3 to 7 carbons. Disaccharide is a combination of 2 monosaccharides. Polysaccharide is a polymer of 5 or more monosaccharides. Monosaccharides and disaccharides are named with the OSE, so if you see OS at the end, then you know that it is a monosaccharide or a disaccharide, and that it was referring to a sugar, so carbohydrates. So hexose is composed of 6 carbons, pentose is 5 carbons, and we do have a lot of hexose carbons, like the glucose, galactose, and sucrose are 6 carbons, but the pentose sugars are found in DNA and in RNA, and they are like ribose and deoxyribose. So some of these are found in other molecules too, but anyway, that's the pentose means 5 and hexose means 6. So you can see here there are 6 carbons, and they have the aldehyde group. And so glucose and galactose are very, very similar. If you look at them, you can't tell a whole lot of difference, except for the 4th carbon here has a little different arrangement. And so you can see that there's a hydrogen on one side and hydroxyl, and this is a kind of like the opposite, hydroxyl on one side and the hydrogen on the other. And so it's this carbon here that differentiates between glucose and galactose. And then fructose has a ketone group, so that is different. So the aldehyde and the ketone, that's going to make this a different shape, and that's going to give it a different function. And these are pretty much the same, but they still have a different shape, so they're still going to have a little different function. So if you have 2 of those, it's called a disaccharide, and this bond that's formed between the 2 monosaccharides, that is called a glycosidic bond. And you see those in the polysaccharides as well. So glycosidic is the bond, and they form by dehydration synthesis. So if you think about this name here, D means to remove, and hydration would refer to water. So when you remove water, synthesis means you build a bond. And so that is indeed what is happening. So we take a hydrogen from one, a hydroxyl from the other, that forms water, and that leaves the space open for this bond to form. And so you can see again, hydrogen from one, hydroxyl from another, and there's your glycosidic bond, and you get water as a product. So here's the polysaccharides. You can have cellulose, which is in lots of repeating chains, has a lot of, it's very strong structure, and then branching chains instead of parallel chains, that's the starch. And again, they have lots of different functions. But cellulose gives strength and rigidity to plants and microscopic algae, so that's in the cell wall of plants and microscopic algae. And it's digestible only by bacteria, fungi, and certain protozoa. Chitin is another, or sorry, we go back here. We have agar. Agar is also a polysaccharide. This is one that's really important, makes our media solid, and it actually is a component of seaweed. It's like a brown algae, and it's from seaweed, but it makes the material able to stay solid even when we reach temperatures up to close to boiling, and then at boiling at 95 to 100 degrees it starts to melt and becomes liquefied. But it's solid as long as it is dissolved in the liquid and everything. It's solid from very cold temperatures up to 100 degrees, and it only melts at like 100 degrees. So it's a natural component of seaweed, and it's a polymer of galactose and sulfur-containing carbohydrates. So that's why it smells when we break down. You know, it's releasing the sulfur when it is autoclaved. So that gives it its nice, lovely smell. Chitin is also found in the cell wall, but it's in the it's in fungi. It's in the cell wall of fungi and the exoskeletons of certain insects and crustaceans. So chitin is in animals as well. Peptidoglycan is what causes or links together and forms the cell wall in bacteria. Lipopolysaccharides, this is the lipid combined with the polysaccharide. That's found in gram-negative bacteria, and it causes fever and shock. So it's kind of it's a toxin. The glycocalyx is this thing that surrounds, it's a polysaccharide, loose or could be very tightly bound around the cell, and it is a way to communicate with other cells. It functions in attachment. It also has receptors, so there's part of cell communication there. Glucose polymers and starch and glycogen store molecules. They store glucose, and they have to be broken down by certain enzymes. And so if bacteria do break down that, this is one of the tests that we do if they produce the enzymes that break down starch. And hydrolysis is the digestion or breakdown of polysaccharides. So hydrolysis is the opposite. They kind of snuck that one in there. The hydrolysis, water. So if you add water, it causes the lysis or the breakdown of the polysaccharides. And so, and this is true for all of the organic molecules. If you have water and probably an enzyme, and an enzyme is usually required, but this causes the breakdown of bonds. So you're breaking bonds. So that's the opposite of dehydration synthesis. So that's carbohydrates.